Welcome to this summary video of the second chapter, Water for Transport. If you haven't gone over the previous videos, I would recommend you do so, but you should still be able to get something out of this actual summary even if you haven't. This video will cover the first 6 videos and the first 6 syllabus dot points of the Water for Transport chapter. So first dot point was identify the forms in which each of the following is carried in mammalian blood: carbon dioxide, oxygen, water, salts, lipids, nitrogenous waste, and other products of digestion. So what you need to know is you need to know these substances that are just named and what form they travel in the blood itself. So we go for each one by one. The first one is carbon dioxide, and it travels in one of 3 ways, either as something called hydrogen carbonate ions. So these are hydrogen carbonate ions. And I've given it number 1 because the majority of CO2 travels as these hydrogen carbonate ions. Remember, that's when plasma reacts with carbon dioxide. When water reacts with carbon dioxide, they form these hydrogen carbonate ions. And that's the number 1 way for CO2 to travel. Number 2 way is for them to attach to hemoglobin. So hemoglobin was in red blood cells. And when they attach to hemoglobin, CO2 attaches to hemoglobin, they're called carbaminohemoglobin. So hemoglobin is by itself, but when CO2 attaches, it's called carbaminohemoglobin. And the third way was that it's dissolved in CO2. So dissolved in plasma. So CO2 is dissolved in plasma, and that just means it's basically swimming in plasma itself. There's 3 ways for carbon dioxide to travel: as hydrogen and carbonate ions, as carbaminohemoglobin, or if it's dissolved in plasma itself. There are 2 ways for oxygen to travel. The number 1 way is the majority of it travels as oxyhemoglobin. So the oxyhemoglobin is referring to if oxygen binds to the hemoglobin, and hemoglobin can be found in red blood cells themselves. So this is the number 1 way, but it can also travel dissolved in plasma. So it can also be dissolved in plasma. So 2 ways for oxygen. We've got carbon dioxide, oxygen, and next is water. Water just travels as water molecules, but most of it is water. So plasma is few salts and other products of digestion dissolved in water. So I think 98+% of plasma is water. Salts travel as ions. These blue molecules or blue ions are supposed to be salts. Salts travel dissolved in plasma as ions. This word here, ions. Products of digestion, so that's all those nutrients that we have to absorb that we need for protein building and for cellular respiration. So glucose and amino acids, they also are dissolved in plasma. So they travel as their own molecule, but they're dissolved in plasma. And then we've got nitrogenous waste. Its form, it travels as urea. So these brown dots, and urea is also dissolved in plasma itself. So we've got 1 more to go, which is lipids. Lipids travel as chylomicrons, and remember why? Because chylomicrons are a protein carrier, and they carry the lipids right inside. And the reason why: lipids are fat, and fat can't dissolve or can't separate, can't mix with water. So the problem is if the lipids were to travel in water itself, it would kind of travel on top, like so, and it wouldn't actually be able to travel itself. It would just be stuck. So by putting them into these chylomicrons, they can travel in the bloodstream and travel from wherever they're produced to where they're needed. So lipids travel in the form of chylomicrons, and they also travel in the blood through those chylomicrons. So that was that. If you have any problems with this one, that was the first video of the Water for Transport chapter. Then you did an experiment as well. You would have looked at what dissolved CO2, so what bubbling into a glass of water, what that does to the pH. So demonstrate the effects of dissolved carbon dioxide on the pH of water. So you would have looked at the pH of water. So you would have had an experiment similar to this. You would have had 2 beakers, and one you didn't do anything with, the other one you would have bubbled into, like so. And what you would have found is that the pH of the one that you bubbled into would have decreased. pH decreased. And remember why? Because the pH in terms of CO2, what CO2 does when it reacts with water, so when these 2 react, it produces hydrogen carbonate ions, which we mentioned earlier, which were these ones. But also importantly, it produces these these hydrogen ions, these ones here. And what they do is they make the water more acidic. So the more CO2 we bubble, which is what we exhale when we bubble into a straw, the more of this we produce, the more acidic the water becomes. So you would have found that in a prac that the pH decreases when you bubble into it. You would have also looked at the white blood cells and the red blood cells. This is first-time investigation using a light microscope and prepared slides to gather information to estimate the size of red and white blood cells and draw a scaled diagram of each. So you would have had a red blood cell and a white blood cell. You would have seen that the actual red blood cell had no nucleus, but the white blood cell had a nucleus. And the reason why red blood cells have no nucleus is because all they have to do is carry hemoglobin, so there's yellow dots in the middle. And then you would have measured them as well, and you would have found that the red blood cells are roughly in that area of 8 micrometers big, or tall. Big is very better. And then you have the white blood cells being roughly 14 to 16 micrometers big, so they're about 1.5 times as big as your red blood cells. So that was our experiment, or that prac you've done in class. You would have also looked at this one, explain the adaptive advantages of hemoglobin. So why is hemoglobin good? The thing with hemoglobin is, remember, we have oxygen can travel one of two ways. Oxygen can either travel dissolved in water or dissolved in plasma, or it can be attached to its hemoglobin molecules. But the problem is not much oxygen actually dissolves. So oxygen doesn't dissolve well. So only about 2% of our oxygen travels dissolved in plasma. The rest of it is in hemoglobin. If you remember why we need oxygen for cellular respiration. And most importantly, we need oxygen to make that ATP through cellular respiration. So if we only had one mechanism to transport oxygen, which would be dissolved in plasma, we would have a lot less oxygen, which we need for making energy. So having a second way means that we can actually pop in a red blood cell into the blood vessel, and this will carry a lot of oxygen. So most of the oxygen, so about 98%, is carried through red blood cells via the hemoglobins. Remember hemoglobin? So hemoglobin, heme stands for iron, and this is a zoomed in hemoglobin. These green dots are the heme groups. That's where oxygen binds to. And the globin was that protein, so that the yellow big thing that the heme groups are in was the globin, the protein. The heme group was the iron, but you have oxygen attaching to the heme group inside the hemoglobin. So that's why we need to have these hemoglobins, because they make us carry much more oxygen, and we need oxygen to produce energy, and also importantly, we need it to grow. So ever since we could do this, we could grow much taller. So beforehand, we were just bacteria-sized, and then now we can grow into human size, or bigger even, because of that cellular respiration. Then we also had analyzed information from secondary sources to identify the technologies that allow measurements of oxygen saturation and carbon dioxide concentration in blood, and describe and explain the conditions under which these technologies are used. So this is 2 parts: identify the technologies and why they're being used and where they're being used. So remember these 2 technologies: the pulse oximeter here, this one. That was the one that you put on your finger. It's like a clip, and it checks the color of your blood. If it's blue, then it means you have low oxygen levels. If it's red, that means you're full of oxygen. That's good. And so that's what it does. This pulse oximeter measures oxygen concentration. And where it's used in early monitoring settings in hospitals. So if you go in a hospital and you have operation, they will usually give you one of those clips afterwards to constantly check on you. So just to check on you. And it comes in very handy because it's easy to do, it's cheap, and non-invasive. So you don't have to have blood taken or anything else. So these are used in early monitoring settings when there wasn't an emergency yet, just checking on you basically. And the other one was the arterial blood gas analysis, this one here. And this one is a bit more involved. So what they do is they take your blood and they put your blood on one of these tubes here. And with that, they check not just your oxygen, but also your carbon dioxide, your oxygen, and your pH. So you can imagine if you get a bad reading from the pulse oximeter, if the nurse or your doctor is alarmed, there might be a problem. Then they'll do this extra test in emergencies to see if what your pH and what your CO2 levels are in your blood, to see if there actually is a need for intervention. So pulse oximeter and arterial blood gas analysis. Pulse oximeter is used in early monitoring settings, and arterial blood gas analysis is used in emergencies. We also have compared the structures of arteries, capillaries, and veins in relation to their functions. So in this one, you had to name the actual structure and how that's related to its function as well. Let's start with the arteries. Remember, arteries carry blood away, so A for away from the heart. They have thick walls, and the reason why is because they have to withstand a lot of pressure. If you remember that arteries were the things that were connected to the pump for the heart, so all that blood that is being pumped out at high pressures is going to go right into the actual arteries. So if they have thin walls, they're going to rupture. So no rupturing happening because of these thick walls, so they can withstand high pressure. And they also have large elastic layers, and the reason why is because they have to maintain a constant blood pressure. And there's usually a heart pumping phase and a heart relaxation phase. And doing hard pumping, the pressure would be high, and doing hard relaxation, the pressure would be low. But to make sure that it's always the same, you have that elastic layer that can always bounce up and down depending on if it's pumping or if it's relaxing. So it's a large elastic layer to maintain blood pressure. Veins, on the other hand, had a thin wall because there was little pressure. Blood was returning. The pressure was already gone. Small elastic layer because the pressure fluctuations were a lot less compared to the arteries. And it had a large bore. The bore was this part, so the opening of the, should use a different color, the opening of the blood vessel. And it was large because it had to return a lot of blood. So all that blood that came back for the veins, there was a lot of it, so it had to have a large bore to return that. Whereas capillaries, they had a very thin wall. So veins had a thin wall, but they had a very thin wall. And the reason why is that because they were attached to all the cells, and you had nutrients that had to diffuse over into cells, and you had to have the waste products diffuse into the blood. If you had very thick walls, that wouldn't be able to happen. Diffusion wouldn't be able to happen. So you have very thin walls. And a tiny bore. So tiny bore, again, refers to that hole, that opening, where the blood can pass through. And it's just big enough for nutrients to exchange. So one red blood cell thick. And the reason why, because all it has to do is it has to supply these cells around it with the nutrients and remove waste products as well. So there's no need to have large bore. It just needs to be large enough to supply these cells around it. So I hope that was helpful. You're going to have the next 6 coming up very soon as well.